Hi everyone, I'm A. Rajarajeshwari from 3rd BSc Biotechnology. I'm studying in Dr. Umyal Ramanathan College for Women. Today I'm going to teach about nuclear magnetic resonance, how it works and what is principle. Let's see. NMR was discovered by Isaac Rabi, an American physicist. Let's see about definition of NMR. NMR, nuclear magnetic resonance, is a powerful analytical technique used in chemistry and biochemistry to study the structure and dynamic of molecules. Basic principle: nuclei with an odd number of protons or neutrons possess a magnetic moment when placed in a strong magnetic field and subjected to radio frequency pulses. This nuclear resonance emitting signals that provide valuable information about the sample. NMR instrumentation contains magnet, radio frequency, sample holder. Magnet. Magnet describes the essential component of NMR machines, a powerful homogeneous magnetic field typically produced by superconducting magnets. Radio frequency coil explains the role of radio frequency coils in delivering pulses to the sample and detecting the emitted signals. Sample holder. The sample containers used in NMR often made of glass and inserted into the magnet for analysis. Chemical shift in NMR. Definition. Chemical shift is the displacement of NMR signals along the frequency axis, measured in parts per million. Factors affecting chemical shift: electron density, magnetic shielding, and neighboring atom influence the chemical shift of a nucleus. Application: chemical shift information helps identify functional groups and determine molecular structure. Chemical spin coupling in NMR. Definition: spin coupling results from the magnetic interaction between neighboring nuclei. Multiplicity: spin coupling leads to the splitting of NMR peaks into multiplets, providing information about the number of neighboring protons. Application: elucidate molecular connectivity and confirm structural details. Advanced NMR techniques: 2D NMR, solid state NMR, 2D NMR. 2D NMR introduces two-dimensional NMR techniques such as COSY, coloration spectroscopy, and HSQC, heteronuclear signal quantum coherence, for more detailed structure information. Solid state NMR, studying solid materials, providing insights into crystal structures and molecular dynamics.
Hello everyone, today we can see Biophysics Unit 5, PCR, DNA sequencing and ELISA. PCR, polymerase chain reaction. It is a technique used in molecular biology to create several copies of certain DNA. It is discovered by Kary Mullis. Its principle is based on enzymatic replication of DNA. These have the following steps: denaturation, annealing, elongation. Firstly, double-stranded DNA is converted to single-stranded DNA and primer is added at 3' to elongate the strand by DNA polymerase to adding a nucleotide. Components of PCR: buffer, primer, DNA polymerase, deoxyribonucleotide triphosphate, Taq polymerase, DNA template. The steps have a specific temperature: denaturation 94 degrees Celsius, annealing 54 to 60 degrees Celsius, extension 72 to 80 degrees Celsius. Application: it is used in medicine, forensic science and research. Second method is DNA sequencing method. It is used to exact sequence of nucleotide or bases in DNA to determine. Maxim and Gilbert method and Sanger method. Maxim and Gilbert method based on chemical modification of DNA. Sanger method is based on enzymatic method. It is discovered in 1977. The Maxim and Gilbert is discovered in 1976 to 1977. Firstly, we can see Maxim and Gilbert method. Cleavage of pyrimidine and purine is a process in that method. Base specific cleavage of DNA by certain chemicals. The nucleotides added up to 500 nucleotides. Advantages. Purified DNA can be read directly. It is used in DNA protein interaction, used to analyze nucleic acid sequence or structures. Disadvantage. Extensive use of hazardous chemicals. Read length decreases from incomplete cleavage reactions. Next is Sankar method. It uses a dideoxynucleotide to terminate DNA synthesis. DNA synthesis have a radioactive substance to yield a series of DNA. It is a process. It is otherwise called chain termination method. Advantages. It have a greatly simplified DNA sequencing. Disadvantage. Non-specific binding of primer to the DNA, affecting a accurate readout. It have a following steps. PCR with the fluorescent chain terminating DDNTP, size separation by capillary gel electrophoresis, laser excitation and reduction by sequencing machine. Third one is enzyme linked immunosorbent assay, ELISA. It is a 3 types. Direct, indirect, sandwich. Direct, it is the simplest format requiring an antigen and enzyme conjugated antibody specific to an antigen. Indirect, 2 steps. First, unlabeled primary antibody bind to specific antigen. Second, enzyme conjugated secondary antibody that is directed against host species of primary antibody is applied. Sandwich. The ELISA measures antigen between 2 layers of the antibodies. Target antigen must contain at least 2 antigenic sites capable of binding to antibodies. Monoclonal or polyclonal antibody can be used as a capture and detection antibodies in the ELISA system. Thank you.
Hello everyone, this is Mridula from BSc Biotechnology. Let's look into the topic chromatography. Chromatography is a technique used in chemistry to separate and analyze mixtures of substances into their individual components. It works by passing a mixture through a stationary phase which interacts differently with the various components, causing them to move at different rates. Let's look behind its history. The term chromatography was first coined by Michael Sweat. This guy in early 20th century filled a glass column with calcium carbonate to separate plant pigments, marking the first recorded use of chromatography. Let's look onto its principle like we do have in our lives. The principle of chromatography is based on the differential affinity of compounds in a mixture for a stationary phase and a mobile phase. It has 3 components: stationary phase, mobile phase, and analyte. The stationary phase is a material that remains fixed in place during the chromatographic process. It can be a solid or a liquid adsorbed onto a solid support. The choice of stationary phase depends on the specific application. Then comes the mobile phase. This is a fluid that flows over or through the stationary phase, carrying the mixture of components with it. The mobile phase moves at a controlled rate. Then comes our analyte, which may be a solvent or a solute depending upon the sample. There are several branches of chromatography, of which we are going to see only 4 of them in deep, which are gas chromatography, HPLC, ion exchange chromatography, and thin film chromatography. Gas chromatography is a technique that separates and analyzes volatile components in a mixture. It involves vaporizing the sample, passing it through a chromatographic column with a mobile phase, and detecting the components as they dilute. HPLC is nothing but a high performance liquid chromatography. Its principle is same as the liquid chromatography, but with only change of high performance mobile phase, high pressure of mobile phase, and a metal column. Then comes the ion exchange chromatography. It is a technique used to separate and purify charged molecules in a mixture. It relies on the interaction between charged molecules and a stationary phase with oppositely charged group. As the sample passes through the column, ions with the higher affinity of the stationary phase are retained longer, leading to the separation. Then comes the thin layer chromatography. It is a quick and cost-effective technique for separating and analyzing mixture. It involves applying a sample to a flat surface with an absorbent material, then exposing it to a liquid solvent. Let's look on to some of its application in brief. It has diverse applications including pharmaceuticals, environmental testing, clinical chemistry, food industry, forensic chemical analysis, biotechnology, and more. It's a versatile tool used across various industry and scientific fields for separation and analysis of different compounds. Thank you.
Hello everyone, I am Batma Malini from Department of Biotechnology. Here I am going to explain about the technique electrophoresis. The word electrophoresis derived from a Greek word meaning transport by electricity. Electrophoresis is a separation of compounds that are capable of acquiring electric charge in conducting electrode. The principle of electrophoresis is V is equal to EQ divided by F. where V stands for the velocity of the molecule, E for electric field, and Q stands for the net charge on molecule, and F denotes the frictional coefficient. These are the materials required for the electrophoresis technique. The gentle apparatus required for this technique is power bank, electrophoresis unit, sample injector, and detector. The gentle method of operation of the electrophoresis technique is saturation of the medium with the buffer after sample application. So next to the application of the sample, electrophoretic separation, finally detection. Electrophoresis is classified into two types. They are capillary electrophoresis and zone electrophoresis. So capillary, it is also known as the moving boundary electrophoresis. First, zone electrophoresis. It involves the migration of the charged particle on the supporting medium. It uses paper, cellulose acetate membrane, starch gel, and polyacrylamide. First of all, paper electrophoresis. Here, it is carried out in a filter paper, and this technique is useful for the separation of the small charged molecule. Next to the paper electrophoresis, gel electrophoresis. It is used for the separation of the DNA, RNA, and protein molecule. Different types of gels are used. So they are agarose gel, polyacrylamide gel, and Sabadex gel. This is the overview of an gel electrophoresis. STS page electrophoresis. So STS page stands for sodium todecyl sulfate polyacrylamide gel electrophoresis. STS is an anionic detergent which binds strongly to and denature the protein. This electrophoresis is used to quantify the proteins, plotting applications, and to determine the molecular weight of the protein. Moving boundary electrophoresis. So this method allows the charged particles to migrate in a free moving solutions without the supporting medium. It carries out advantages like the samples can be detected in very minute concentration, and disadvantages is it is very costlier. And this type of electrophoresis technique is used to analyze the complex biological mixture. Capillary electrophoresis. Between two buffer reservoir, capillary tube is placed, and after applied the electric field, separation depends on the electrophoretic mobility and electroosmosis. So this technique is used without the gel. Isotachophoresis. So iso means equal, tacho speed, and phoresis means migration. This technique is useful for the separation of smaller ionic substances, that is the separation of the small anions and cations. The factors that affecting the electrophoresis are sample. The sample includes charge, size, and shape. Electric field. Electric field includes voltage, that is increase in the voltage, increase in the rate of migration. If current increase, it also leads to the increase in voltage, and which leads to the increase of the migration. So finally, resistance. Resistance increase, the migration decreases. Buffer. Buffer includes ionic strength and pH, whereas in ionic strength the rate of migration increases in high ionic strength, and in pH ionization of the organic acid increases as pH increases. Supporting medium. Adsorption and electroendosmosis. Absorption. Reduce both rate of migration and resolution of the separation of the molecule. Electroosmosis. Example, paper agarose. It will accelerate the movement of the cations but retard the anion movements. Finally, applications of electrophoresis. So electrophoresis is useful in the DNA sequencing, medical research, agriculture testing, and in food industry. Thank you.
Hi everyone, I'm A. Radhanadishwari from 3rd BSc Biotechnology. I'm studying in Dr. Romeo Ramanathan College for Women. Today I'm going to teach about autoradiography. Autoradiography is a technique using X-ray film to visualize molecules or fragments of a molecule that have been radioactively labeled. For example, analysis of a length and number of DNA fragments after separation from one another by a method called gel electrophoresis. In autoradiography, the specimen itself is the source of radiation, which originates from radioactive material incorporated into it. This technique detects samples in two ways: direct autoradiography, indirect autoradiography. Direct autoradiography. Direct autoradiography is suited for the detection of weak to medium strength beta emitting radionuclides, for example tritium carbon 14. In this technique, the sample is placed in direct contact with the film. The radioactive emission results in the development of black areas. Indirect radioautography, detection of highly energetic beta particles, for example phosphorus 32, is not done suitable by direct autoradiography because this emission passes through and beyond the film, and a major part of the energy gets wasted. In this technique, the beta particles energy is first converted to light by a scintillator, which then emits photon and exposed to photographic emulsions. Direct principle. Autoradiography is based upon the ability of a radioactive substance to expose the photographic film by ionizing it. In this technique, the radioactive substance is put in direct contact with the thick layer of a photographic emulsion. That photographic emulsion having a gelatin substance and a silver halide crystals. The emulsion differs from standard photographic film in terms of having a higher ratio of silver halide to gelatin, and then for several days it kept for a proper exposure. The silver halide crystals are then exposed to the radiation, which chemically converts silver halide into metallic silver. That metallic silver giving a dark band, color band. Then we can resulting radiography and viewed. And the resulting radiography is viewed under the electron microscope or electrophoresis or any method like a digital scanners, etc. Methodology. The radioactive sample is covered with the photographic emulsion. That photographic emulsion is having a silver halide and the gelatin. The radioactive part of the sample activates the silver halide crystal nearby, and then the result in reduction of silver ions to silver atom, leaving dark color bands. Then the slide is washed away by the fixers to get insoluble silver atom only. The autoradiography can further be viewed under the microscope. Method. The autoradiography has two types: macro autoradiography, micro autoradiography. The procedure of macro autoradiography. The radioactive tracer is introduced into an organism by injecting or injection. The individual sample of tissues are removed and pressed directly against X-ray film for several days to expose the film. The film is then developed and viewed by microscopic. The micro autoradiography. A refinement of the methodology is called micro autoradiography. This was developed for study of subcellular structures. Example: DNA. Micro autoradiography procedure. The cells are first incubated in nutrient solution containing molecules. That molecules have been labeled, usually with a radioactive carbon, tritium or phosphorus. After a period of incubation, some cells are transferred to a glass slide. The slide is dipped into a liquid photographic emulsion containing light-sensitive silver bromide. The slide is then placed in dark for several days for radioactive decay. The beta particles from tritium cause the photographic emulsion to become exposed. The slide is then washed away by fixers to get insoluble silver atom only. Finally, cells are examined with the microscope. Applications of autoradiography. To find and investigate the various properties of DNA. Find the location and the amount of particular substance within a cell, including cell organelles, metabolites, etc. Tissue localization of a radioactive substance. To find the site and the performance of targeted drugs.